How to use a hemocytometer.
In this video, we will demonstrate how to use a hemocytometer to determine the cell density of a culture. At first, a hemocytometer may look like other slides you've used to observe microbiology under the microscope. However, if you look closely, you will see its unique features. First, take a look at one of the two sampling areas and notice the loading area, which is used to load your sample. Next, look at the detection area, which is used for counting cells in your sample. Now, check that you have all the supplies you need to determine cell density. These include a disposable C-chip hemocytometer, a micropipette for measuring 10 microliters with sterile micropipette tips, a clicker, a microscope capable of 40 times magnification, your cell sample, and a lab notebook and pencil to record the results and make calculations. The first step is to load the sampling area of the hemocytometer with 10 microliters of your cell sample. Adjust the setting on your micropipette to read 10 microliters. Now, vortex your sample for approximately 3 seconds before loading it. This is important to ensure that cells are suspended in the solution rather than concentrated in one area. In order to approximate cell density, it is important to load a representative sample. If you do not have a vortex on hand, you can also gently pipette the sample. Next, using a clean pipette tip, draw up 10 microliters of your sample and expel it into the sample loading area. Once the hemocytometer is loaded, it is time to count the cell sample. Do not wait too long before counting, or the sample will start to evaporate, pushing the cells together and changing the density. You will know this is happening if your cells begin to move under the microscope, or if you see air bubbles. Stop counting, load the second sampling area, and start again. Check with your teacher to see if you need to dispose of your hemocytometer in a biohazard bag. This can be important when working with cells. Find the grid on the 10 times magnification and center the microscope on the top left quadrant. Switch to the 40 times magnification and phase 2. Now we will center the microscope on the top left square in the upper left quadrant. Click the counter for each cell in the square. Then scroll to the square to the right and continue counting the cells, focusing on one square at a time. Once you have counted all 16 squares in a quadrant, record the number on your clicker in your lab notebook. Reset your clicker and move to the next quadrant. Rotate through all 4 quadrants clockwise until you have recorded the number of cells in all 4 quadrants. Note: cells on the left and top sides should be counted as part of the square. The cells on the right and bottom sides should not. Also, if you do not have a clicker, you may ask your partner to tally the cells as you count. Notice that the hemocytometer has 2 usable sampling areas. If your cell density is low, it may be necessary to count 2 samples. First, average the cell counts of the 4 quadrants. If your sample was diluted, divide by the dilution. Next, convert this average to cells per milliliter, the unit typically used to record cell density. To do this, divide the average number of cells per quadrant by the volume of each quadrant. Density equals average cells in a quadrant over volume of a quadrant. Remember, each quadrant is a millimeter by a millimeter square and has a depth of 0.1 millimeters. So the volume of each quadrant is 1 millimeter by 1 millimeter by 0.1 millimeter, or 0.1 millimeter cubed. Divide your average cell count by 0.1 millimeters cubed. Now, convert millimeters cubed to centimeters cubed by multiplying by 1,000. Centimeters cubed can be converted to milliliters by remembering that 1 centimeter cubed is equivalent to 1 milliliter. So now we have average of quadrant cell counts over 0.1 millimeter cubed times 1,000 millimeter cubed over 1 centimeters cubed by 1 centimeter cubed over 1 milliliter equals cells over milliliter. Now you have your cell density.
Yay!